scientist and a radiological safety officer IRI, Dr. Bhupinder Singh sir. The research area of his interest are radioisotopes and radiation applications, mineral nutrition of crops, air pollution monitoring and its impact on crops. He has published more than 150 research papers in peer-reviewed journal with very high impact factors. And he was honored by various awards, Biocost Fellow, and he was visiting scientist at Germany. So with this note, I request my coordinator, Dr. Ranjit Ranjan Kumar, to kindly felicitate sir with flower.
This is welcome and hope that next time I welcome him as a director. Yes.
So the title for today's talk is on application of radioisotopes in deciphering inome interactions and source sink relationships in plants. So I request and invite Dr. Bhupinder Singh sir for his talk. Welcome.
Thank you, Suneha. Thank you, Ranjit. Thank you, everyone. I hope, like the morning lecture, you will also be getting some good ideas from this lecture. Because these are specialized lecture, I would call it like the radiation applications. I am going to talk about the radioisotope application. And when we in the morning, my friend Dr. Jambulkar talked about radiations, radioisotopes are the source of radiations. Okay? So, these radiations which are emanating, which we are exploiting, essentially are coming from radioisotopes. So, you can see my talk, application of radioisotopes in deciphering the inome interaction and source sink relationship in plants. So, three keywords that are mentioned here is one is the radioisotopes, second is the inome interaction. Inome, you guys already might have been told about it. It is the micronutrient and trace elements, mineral nutrients that are there in the tissue. Complete profiling of the mineral nutrients, macro and micronutrients in the tissue. So, whether there are interactions, so I would simply say that Dr. Ranjit is excellent in the class, okay, till Suneha joins. When Suneha joins the class, she becomes excellent. So, this is the interaction. Do not take these words these are only analogies, these are not to be taken on face value. But the point is that when one nutrient alone is present, its behavior will be different, but when it is in the presence of multiple other nutrients, there will be competition, there will be synergies, there will be antagonisms, all of which will influence not only the uptake, but also the translocation and various other physiological parameters to affect the enhancement of nutrient in your plant or the final produce. So, this is one aspect that we are going to talk about inome interactions, and source sink relationship is one. Source sink relationship, essentially we talk about the carbohydrates or the carbon metabolism of the plants, but you can also revisit source sink relationship in terms of the nutrients. When I was a child, I was a sink to my parent. Now I am grown up, I am earning, now I am a source to my kids. My kids now have become the sink. This is how, when the leaf is young, it is a sink. When it matures, it becomes a source. Okay? So, all these aspects, how the relationship, dynamics of source sink relationship, it changes during the course of plant growth, we will be talking about. Next one, please. Do I have to do it on my own? I think it's there. Okay. Thank you. Yeah. Huh? You can do it from the keyboard. Someone can sit there and do it from the keyboard. Okay. I'll be happy if when I say next, press the keyboard. That's better. Okay. So I don't know whether my friend, I was a little late in the morning lecture whether Dr. Jambulkar talked about the concept of radioactivity, or he directly started with the role of radiations fermentation breeding. We know that atom is the basic entity of any matter. And what is atom? Atom is having a nucleus which is having protons and neutrons, and there are electrons that are revolving around the nucleus. So when the number of protons is normally equivalent to number of electrons, and it is the number of protons plus the number of neutrons that make up the nucleus constitute the atomic mass, and when we talk about the number of protons alone, they are the atomic number. And isotope of an element, they have the, what are isotopes? They are the isotopes are the elements with same atomic number but different atomic masses. We have been reading it throughout. When you talk about carbon, the atomic number remains 6, but the atomic mass changes from C12, C13, C14. And if where C12 is a natural isotope, C13 is a stable isotope, C14 is a radioisotope. So the concept of radioactivity, when we talk about, it is the ratio of the neutrons to protons in the nucleus that determines the instability of an element, of an isotope. And if something is unstable, I just take analogy of an advertisement that came a few years back where a saint was eating some biscuits. They were probably trying to showcase that the biscuits are very low in calorie and it will make you lightweight. So what they showed is that someone was eating, the saint was eating biscuits, and then he was rising above the ground. With each biscuit he was eating secretly, he was rising above the ground, and the crowd there, the bhaktas, they were appreciating, thinking that it is something his grace, he is a very godly person, that is why this is happening. Now point is, if someone rises above the ground, if you energize an element, the atom, the electrons move from the lower energy level to the higher energy level, but those higher energy levels are not there static or the ground state. So slowly and gradually, if I throw a ball up in the sky, at one stage it will reach, but that is not its normal stage. So what it will do is, it starts slowly coming down. And when it starts to come down, when the electron which has been with energy, it has been shunted from its lower energy level to the higher energy level, which is not its ground state. So slowly it tries to come back to its original level, releasing the energy, and this energy can be in the form of radiation, which we determine or measure and exploit for our research activities. So when you talk about the nuclear and allied techniques, we have about 8 nuclear power plants already running, and it is an indefinite source of power, indefinite source of power. It is unlike when you are exploiting the natural resources, which are limited, restricted. This can become an unlimited source of power. Nuclear tools, nuclear energy, if we are having thorium, uranium, then these can become unlimited tools of power. We have the nuclear powered submarine. So these are some of the examples where the nuclear energy is also used, the radiations which he didn't talk about because Dr. Jambulkar was talking about its application in the agriculture. You can also use radiation for creating different colors of the stone, the diamonds. If I have some glass pieces and I radiate it at different level, which probably if you come to my lab, I may have some bottles of glass which we radiated at different levels, and you can have a gradation of coloring from the white to the darkest of the color, which people use it, and these are almost permanent color. They will be there for 25 years, and 25 years is a good period. You will either lose that diamond stud, or you will sell it, or you will not will be interested to revere it even after 25 years. So, idea is that the tools are immense. They have very high, immense applications. So, the advantages with radioisotopes are that they can be very easily traced and measured in minute quantities, one. Second, the situations are uncomplicated by the presence of larger pool of ions. Now, point is, you are having, say, I am interested in potassium. I want to know what is happening to potassium in the soil or in the plant system. Now, potassium is a lot in my soil. Nitrogen is a lot in my soil. Phosphorus is a lot in my soil. So, if I want to study the mechanism by which the potassium, sodium, or any other element is altering or affecting or competing, what are the mechanisms? I can use the radioisotope of that, and that situation will not be, even if you have one atom, radioactive atom of sodium in a pool of other sodium on a salt soil, NaCl treated soil, you will be able to identify that trace. You will be able to identify that radiation, which is signal which is emanated by one atom. So, point is, if you have thousands of pool, millions of pool, 1 in million, 1 in thousand, 1 in billion, you will still get the signal. So, situations are uncomplicated by the presence of the larger pool. This is important. These are the common radioisotopes. I will just say sodium 22 is normal, sodium 24 is radioactive. There are 2-3 things which are written here. One is the name, other is the symbol, the other is the decay type. I told you, when the electrons are coming down, they will be releasing the energy, which will be in the form of gamma radiation. If there is a nuclear disintegration or decay, there can be a release of an alpha particle. If the electron gets, instead of going from the lower energy level to a higher energy level, if there is a, what word should I use? There is a ejection of the electron. If there is an ejection of the electron from the atom, then you have a beta particle decay. So, the modes of decay, 3 modes are there. One is alpha, other is beta, third is gamma. I will not go too much into it, but just imagine alpha is having the highest of mass, it is an helium atom. Beta is an electron which has less of mass. Alpha is positively charged, beta is negatively charged, gamma is neutral, it is a radiation. So, alpha, it is heavy, okay. It is having more energy. Suppose you have 3, alpha, beta and gamma having 1 million electron volt of energy, okay. Alpha is heavy, beta is lesser than or less than alpha, gamma is least. So, if I am having the same energy and I am throw 3 shot put balls alpha, beta, gamma over a distance, which of these 3 balls will travel more distance? Gamma will travel more distance because alpha is heaviest. With the same energy that I use, same 1 million low electron volt of energy, it will come to rest by after traveling the shortest of distance. So, that means it is traveling shortest of distance, means when it is coming to the rest, it will lose all the energy that is associated with it. So, when it is losing all the energy, there is a linear energy transfer, energy lost per unit time, unit distance. It will be maximum for alpha and least for gamma, because gamma is traveling large distance. So, it is losing, and it is neutral, charge neutral, so it is not interacting. Alpha will be interacting. If it is at positive, it will be interacting with negative charge and getting quenched. So, that is why we use gamma as a sensor, because even if I have a material, gamma can travel to the and give me a good signal, okay, not the alpha, because alpha or beta can be cut by, alpha can be cut by a sheet of paper or a gloves if I am wearing, alpha can be cut by that, beta can be cut by a plastic sheet or a fiberglass sheet, plexiglass sheet if I am having in front of me, it can cut that, but gamma can be cut by only lead shielding. Okay. So, this was not related, but you should know it. And then the energy, and the third is the half life. The energy which is associated with different atoms is different, different elements is different. The last one is the half life. This is important. Half life means how much time it will take for the energy of an element to become half. Okay. Energy will remain the same, but activity will come down. If I am having energy is characteristic of an element, it is a characteristic of an element. Based on the energy, I can identify, there are radionuclide identifiers and I can identify the element. But the activity, if I am starting with 1 curie of activity, 1 curie of activity will become 0.5 curie in that much of half life. 15 hours of half life. Now, just have the same analogy. If 1 curie is becoming 0.5 curie in 15 hours, and in the carbon 14 it is taking 5730 years to become come down from 1 curie to 0.5 curie. What will be the rate of release of energy over time? In which of these two cases it will be more? One which is taking 15 hours or one which is taking 5730 years? Tell me please. Carbon is taking 5730 years for its activity to come down from 1 curie to 0.5 curie. Sodium is taking 15 hours. So, in which of these two cases the release of energy or the signal will be more? Sodium. Okay. So, if I am carbon, that is why, because of its long half life, carbon is used for carbon dating purpose. Okay. And suppose I have to work with sodium, I am working with sodium. So, my half life is 15 hours. So, that means this half life tells me that how long I can continue the experiment, because with each half life the signal which I am getting is going to reduce. Okay. So, I need to finish my experiment within 6 half lives to get a tangible signal which can be detected by my machine. Next one. So, this is the different nutrients. I will not go into it. You all know macro and micronutrients the plants need. Macro and micronutrients, they need water also, and if that is not there, you have a lot of visible deficiency symptoms. You can also, apart from the visible deficiency symptoms, you can go in for the nutrient analysis in the plant, or you can also go in for the biochemical markers. You can use radioisotopes for understanding whether the uptake process of uptake is active, what is the interaction between the ions, what is the efflux, is there any efflux of the nutrient if you are losing an element, whether there is any effluxes taking place. You can also find out whether you are having the foliar uptake of an element or not. So, all these things you can detect using radioisotopes. So, these are some of the things which I wanted to say, that there are 2-3 things where the ion interactions, the anion interactions can get affected. One is the content and availability. These are the physiological, because there are 2 things which you should learn. One is the total content of an element in the soil. Second is the available content of the element in the soil, of the nutrient in the soil. These two are 2 different things. Soil scientists, they know, they always measure total and available. Physiologists, we measure what is taken up by the plant. And there are interventions which are taking place not only at the soil level because of the microbial populations, or through the root exudation by the plant, which are modifying the rhizosphere in a manner to bring the unavailable form to the available form. So, these two are, suppose we take an example of zinc. Out of the 25 kg of zinc sulphate that I am adding, only 400 gram of zinc will be available to the plant. So, that means there is a lot of zinc in the soil, but it is not available. So, content is there, availability is less. So, if we keep on applying year after year, of course, the recommendation is only after 2 years you should apply. But if we keep on applying year after year, what is going to happen is that you are going to have a buildup of an element in the soil. So, we need to look for the types that are able to utilize the unavailable form of nutrients. They are there, but they are not available. How we can modify the rhizosphere in a manner, or how we can utilize the total into the available. This is one aspect. Rooting characteristics, you heard it in the morning from Dr. Jambulkar, how the roots can play a major role in determining that how much of the soil volumes will get exploited. And it is not essentially the depth. Depth is important when it comes to drought, because you need to go deeper and deeper to search for water. But if it is any other nutrient deficiency conditions, not the depth, but the linear or the horizontal proliferation is also important. So, what the few characteristics of the roots that we talk about is the root surface area, okay, and the root diameter. It has been shown in the case of zinc that if you are having less than 2 mm of the diameter roots, the variety is going to be efficient in uptake. And when it comes to root exudation as a phenomena, which is important for modifying the rhizosphere, it is the number of root tips that are there, because exudation will essentially coming out from the root tips. It is the root tips that are important. So, a variety which is having more surface area, higher surface area will lead to a higher uptake. Imagine 1 gram of root divided into 10 bigger thicker pieces and 1 gram of root divided into 200 finer pieces. They are having a larger surface area to exploit larger soil volumes. So, this is one characteristics. Then is the uptake kinetics. You are biochemistry students, you all know we talk about Km, Vmax and Cmin. Km is the minimum, Km is the affinity for the substrate, Cmin is the minimum concentration at which the uptake will begin, and Vmax is the maximum rate at which the uptake will occur. All these phenomena can be understood with certainty using radioisotopes. You have the translocation. Once it has been taken up by the plant, whether there is translocation taking place or not, or whether it is retaining it in the roots, if you are taking up but not translocating to the shoot, it has no meaning. Not only the nutrient in question should be taken up by the root, it should not only be translocated to the shoot, but it should also be translocated to the grain or the fruit or the economically important edible part. If we want to do justice with our theme. Then is the retranslocation. I took an example earlier that a younger boy is a sink. When he gets older, he is a source, and when he becomes still older, on the verge of No one parts with the income. If suppose I have earned, at what time do you think I will give it to my kids? Not now. When I know that okay, I am going to die in a few years, then I will say okay, I will write my property to the, not to my son. Will I give to my son or will I give to my daughter's son? I will give it to my daughter's son, because for parents, grandparents, it is the grandchild who is more affectionate than the own child. It is not that the child does not love, child also loves the parent, but the grandchild cannot think he is very pure at heart, he does not have any problems, he does not have anything, so he will be without any tantrums, whereas elderly person will be thoughtful, logical, and he will only also sometimes be questioning the father. So, father will give the income to the grandchild. So, retranslocation is a process where a leaf during senescence will transport the nutrients that it is having to the youngest growing leaf, so as the future of the youngest leaf is secure, and youngest leaf becomes full grown. And these phenomena, efficiency of retranslocation is also important for the elements that are least mobile in the plant. One, if something is not mobile in the plant, the deficiency symptoms will occur in the younger leaves. If something is mobile, deficiency symptoms will occur in the older leaves. Okay. So, this is one aspect which we understand is also important, retranslocation of the nutrients, so that the future of the youngest, and particularly important under deficient condition, when there is a marginal soils are there, you do not have something which the plant can take it from the soil. There is a deficiency, so where from the nutrients will come? They will be coming from the leaves that are already there. Then is the utilization efficiency. I also eat 2 chapatis. Now Ranjit again joins me, so I will take his name. Ranjit also takes 2 chapatis, but my weight is 90 kgs, his weight is 60 kg. So that means my utilization efficiency is more. So it is not what you uptake, how you utilize. I think there will be, there are already, you might have been told about nutrient homeostasis, how the nutrients not only are partitioned between the tissues, root and the shoot tissue or the grains, but also how they are at the cellular level, how they are partitioned, whether they will be absorbed or retained at the apoplastic region, or they will be remaining in the cytosol, or they will be pooled, they will be sent to the vacuolar pool. How the cellular partitioning is taking place is also very, very important. So utilization is very critical, because these nutrients are either, they are the cofactors, or they are associated with, they are either propelling certain enzymes, or they are they are having a structural role in the enzyme systems, very, very critical, in both either structural or functional role, they are very critical. Okay. So how these are partitioned so that the various physiological functions are optimized, particularly under the deficient conditions. If something is available, I do not have to think of the strategies, but if something is less, I need to think of the strategies that will help me optimize different pathways so as I complete my life cycle happily. Then is the rhizosphere modifications that we talked about, the root exudation will be there, and how it is important we are going to see. And then is the biofortification aspect, which Dr. Jambulkar also talked about, that it is the nutrients in the edible part that is of consequence rather than the nutrients in the vegetative part. Morning he gave a very good example of mustard. If you have a profuse, and he was also sharing with us later, that if you have profuse vegetative mass with very poor yield, farmers do not accept it. Farmers will be happy if you have less vegetative mass. Less vegetative mass also, you can enter the field, you can do all the agronomic practices, and you have good yield. all the agriculture agronomic operations also become very easy if the crop is short. This is a very good example. You can see the competition between the cations, how in this case only magnesium 28 is used, radioactive magnesium, and this is to exemplify the cation competition that is taking place. You can very clearly see, and what I am measuring here is only magnesium activity. I am only measuring the uptake of magnesium, how the uptake of magnesium is getting affected in the presence of other cations. So, in first case I am having only magnesium, in the second I am having magnesium chloride plus calcium sulphate, I am adding one cation more, calcium. In the third case I am having magnesium, calcium and potassium, 3 cations are there. So, how the uptake of magnesium is getting affected in the presence of other cations. This is very easy to detect, and it is possible only with the use of radioisotopes. You can very clearly see when magnesium alone is there, its uptake is units, I will not read it, but the uptake is almost, it is more in the root and less in the shoot. But you compare it with adding one calcium, the uptake, total uptake also reduces, the translocation of magnesium from the root to the shoot also reduces. Third case, if you are having one more element added, cation added, potassium, from 165 magnesium ions, it goes down to only 15. So, 10 times reduction is there in the root. If I see the total, it is almost 220, 245, and it is only 24, almost 10% uptake. So, you can very clearly see that not only the two things which we can infer from this is that not only the cations, presence of the other cations will affect the uptake of the main cation. If potassium is there or calcium is there, magnesium will not be taken up. So, despite, what does it tell us? It tells us that if you are soil, if what I can do as a soil scientist, what I will do is I will take a soil sample, I am having deficiency of magnesium in the plant, I will take the soil sample, I will analyze for magnesium, I will say I do not know whether my analysis is faulty or what, I am having lot of magnesium in the soil, why the deficiency, the hell is coming? This is because of the cation competition, that is, then you should analyze the presence of the other cations in the soil, whether potassium is more, ammonium is more, sodium is more, what are the different cations that are there? You analyze that, and if you have more of those cations, calcium is more, if more of those cations is there, then magnesium, despite its presence in the soil in larger quantities, it will not be taken up by the plant. Not be taken up by the plant. And here this slide also tells us, this is it's all about inferences that you can draw, not only uptake but also the translocation of the nutrients will be affected. You can see 165, 88, 115, 25. Not only the cation competition is not only influencing the uptake but also the translocation of the magnesium, because we use magnesium, so I will be drawing inferences for magnesium. It will only also be affecting the translocation of the magnesium from the root to the shoot. This is another one which always I like to tell you guys. Here you can see what we did is we are comparing soil with water culture and having phosphorus P32. And why we do is that many times we are lost, we do an experiment with the soil and then we find that uptake is not there, and when you see it in the literature, someone else has shown that there is a lot of uptake is there. So, just look at what the methodology, which methodology he used, whether he used hydroponics or water culture or he also used soils. If you use soils, then you can relate it with the other cations or anions that are present, or if the methodology he used is hydroponics, you can very clearly see that if it is a hydroponic or if it is a soil, water culture and soil, in water culture in 60 hours not only the root activity is translocated to the shoot, but in water even in the soil it is even the roots, they are not taking up that much of P32 as compared to so these signals, they are very radioisotopes are very important for understanding such phenomena. So this is you can see the activity in the soil, you can see here this is in the uptake by the soil, relative counts that you get it in the soil, whereas in the water culture they are increasing. So these kind of variations, so always I advise is that you guys should keep a note, make a note of the methodology. Just do not compare if you are getting more protein content and you find, oh, the other gentleman in the literature, sir, it has shown lesser of it. You just check your methodology. If your methodology is fine, then you rely on that. You take it as a right value. You check your data, check your methodology. Don't get what do you call bogged down by something which is there in the literature. Then look for his methodology, counter that methodology, counter that, have an argument for their result, counter argument for their result, but trust your results first. I'll give you an example. See, my student in 2007, Pratibha, she later on went to do PhD in IASC. MSc, she found that the the root exudates of wheat can help improve the micronutrient content of brassica or dicot. So, root exudates of wheat, if suppose you can have it, either isolate the root exudates of wheat, apply it in the rhizosphere of the dicot, or you can go for intercropping of these two lines, and you find that the brassica is also getting enriched, the seeds are getting enriched with the micronutrient. We did not publish, so we probably, the literature at that time was silent on that, she did not publish, and in 2018-19 now the papers are coming that root exudates of monocots can help improve the uptake of nutrients in the case of dicots. So, you should have courage, and courage comes when you rely on the data. And who can give that much of confidence? You as student should give confidence to your supervisor, and that will come when you do it correctly, you appreciate your work first. Okay. This is regarding the semen. See, I have already spoken about it. I will not getting in deep into it. This is using zinc 65. How we can use to find out the semen, Vmax, and the Km. Okay. I'll just share with you the depletion, nutrient depletion method. Suppose they have kept water here. This is water culture. Okay. I add radioactivity in this. Suppose I am interested in phosphorus dynamics. So, I add P32 at the rate of 2000 becquerel counts per ml. Suppose I am having 250. So, I add the activity at the rate of 2000 becquerel per ml. That much of activity I add in water. Then I add take the plant, whether it has been raised in any condition. I am interested what will be the uptake dynamics of that. So, I put that plant into it. I conduct a short term experiment. Initial count, I know it is 2000 becquerel per ml because that is what I added. That is a 0 time count. After 5 minutes, I take some alicot from here, replenish it with the other water. 100 microliter I collect, take the counts in that. 10 minutes, take a count. 15 minutes, take a count. Up to 1 hour or 2 hours, I can conduct this experiment and measure the count. If initially, and if I am having 2, 3, 5, 10 varieties, this is nutrient depletion experiment because the plant will be taking up the nutrients in the solution will get depleted over time. And it is the rate of depletion you can calculate from, if you are having uptake within 2 time frames, you can calculate the rate at which the nutrients are being taken up by the plant. Later on, when the experiment is over, after 2 hours you remove the plant, cut the root and shoot, measure the counts in that, and you get the one which I showed you already, the counts in of magnesium in root and shoot. You can measure that. So you can know the translocation, you can know the uptake, and you can know the depletion, the kinetics you can work out. If you are initially you are having high uptake, okay, of the nutrients, if I am measuring with this, I am getting a higher uptake, and it continues to remain high even at, because what will happen? Say in 1 hour the concentration which I took 2000, it will become 1000. And even if my variety is able to take up nutrient from that 1000 ppm, 1000 becquerel count, when the concentration of the nutrient in this is low, which means that it is having not only the low affinity transporters but also high affinity transporter. Low affinity transporter is which will be active at higher concentration of the nutrient, and high affinity transporter will be active at low concentration. If you find that it is having a high activity but after a while it is coming down, that means this variety is probably not bestowed with an active high affinity transporter. So you can calculate this, and if you continue this experiment, you can also find out the minimum concentration at which it is showing the uptake. Cmin you can find, Vmax you can rate, if you find the maximum rate, you can find at what period it gives the maximum rate. So all these kinetics you can find out using the nutrient depletion experiment. This is another experiment, not ours, but I used it just to show that here are 2 plants which are minus iron and plus iron. You can see it from the chlorophyll itself. One is darker in color, other is pale. And if I supply iron 59 Ma, Ma is a mugenic acid. Okay, this is a chelate. Iron is less mobile in the plant, it is least mobile rather in the plant systems. So it is, you always supply it in a chelated form. So when you are supplying it as a chelate, the movement of the chelate, radioactive chelate in the plant, you can see I am not hungry, I was grown in iron plus conditions. So when you supply iron to me, if you are putting it around me, you will have it around my body, but my body doesn't need it, so it will not be getting absorbed by my body. But under iron minus conditions, the situation is different. Not only the iron will be taken up by the root, but also will be translocated. So this you can find out using. This is another example of, we worked on zinc, so most of the examples you will have it on zinc. So this is the retranslocation, how the senescence, during senescence, the older leaves, they transport the nutrients to the youngest leaf, and this is also a very, very important strategy for improving the nutrient use efficiency, okay, physiological efficiency. This is another example how using carbon C14 carbon dioxide we can find out. It's a rapid method for finding out the root exudation. Root exudation, the organic acids, they are all carbon. Proteins, they will also have carbon. So what all is released, we are talking about the total exudation. So what we do in this case is, we raise the plant, all of you see here please, we raise the plant and we put them in a chamber and we release in the chamber under control condition, we release C14 carbon dioxide from labeled sodium bicarbonate, that we get it from Board of Radiation Industrial Technology, Bombay. So if I am having sodium bicarbonate, I add HCl into it, what I will get is NaCl plus CO2 plus water. This CO2 will be C14O2 will be getting all around in the environment of the plant and will be taken up by the plant. and when it is taken up by the plant, if I later measure the exudate, or if I am raising it in the soil, and if I water or irrigate the soil, okay, I drain the soil, I measure the radioactivity in the drainage, whatever total I will get, the total radioactivity I get it in the drainage water, it will be essentially that of the total, it will give me an idea about the total root exudation. So, if I want to follow this strategy of total root exudation as a parameter which is likely to affect or influence and modify the rhizosphere in a manner to improve the nutrient use efficiency, this is the best method. You can define the efficient and non-responsive, efficient and responsive, you can categorize them based on with the relative biomass that they are producing and what is the level of C14 exudation by them. So, this is also very important technique that we exploit. This is also another one is the phytosidrophores mugenic acid that I told you, these are phytosidrophores, these are non-proteinaceous amino acids or the chelates, particularly in the monocots, okay, and they have a role in uptake of not only zinc but iron, manganese, copper, and most of the nutrients when we talk about, we do not talk about nitrogen in the plant, we do not talk about others, but we talk about the micronutrient deficiencies which are critical. So, here we have the root exudates which are not only taking up, and this strategy why we are talking, because I told you, gave an example, zinc is in plenty in the soil, but it is not available. So, it is mobilizing the unavailable or the immobile form of the zinc in the soil, it is making it mobile, forming a chelate, and then uptake is as a chelate, transport is as a chelate, and as a chelate itself it is transported to the grain, and this strategy is helping to improve the grain micronutrient content. This I must tell you, this is a experiment which we designed. This is a mixed culture experiment. We had the bread wheat which was producing more of the root exudates, the durum wheat which was producing less of the root exudates. So, we had them in a condition of mixed culture condition where we had one plant of bread, one plant of durum. Assuming that the root exudates in the rhizosphere under the mixed culture will be in between A and B. The availability of the root exudates will be, the condition will be between the bread and the durum. So, if the durum is able or getting benefited by the root exudates from the bread, okay, that means it is having, we can, if we improve the root exudation of durum wheat, we can make it as efficient as bread wheat. So, idea of doing this was, then what we did was that we provided zinc resin in a dialysis tube and we hung it in between that. So, it is already bound resin, zinc bound resin, sardolite key. This is the resin which is specific for zinc. We have it in a dialysis. So, PS, what it will do, the sidrophores, what they will do is, they will be moving through the dialysis tube, immobilizing or removing the zinc from the bound form, coming out as a chelate and then being taken, and still if you find an increase in the content, that means you are getting benefited. Okay. So, this is the result. What we got is that you can very clearly see is BB is bread, BK is K is here the Kundru variety that we use. So, it is a durum type. You can see the color, the KK is total pale, BB is total green, and in the case of B and K, you still have, you very clearly see that this is getting benefited. Okay. Then another experiment which we did was that when you are supplying zinc 65 bound chelate, DMA is deoxymugenic acid, and I am supplying zinc 65 bound chelate, same concentration to these two plants, bread and durum, and if still they are able to take up at the same level that means the inhibition or restriction or limitation is operative at the level of release, but not at the level of uptake of the bound form. So, if very clearly you can see here, since bread and durum under zinc minus conditions, they were equally able to take up the zinc 65 bound chelated zinc 65. That means the inhibition or limitation is not occurring at the level of uptake of the chelated form. So, if we improve the release of the root exudate in the case of durum, it is more likely to get benefited. Okay. So, some of these experiments, these are not possible to do it without radioisotopes. This is another one very beautiful strategy which I love to tell you guys. I always wish to develop methods. This is one method which is very important, is the dual labeling technique. Dual labeling technique is when you have, there is a chelated nutrient. So, how far you label the carbon of the chelate and the element nutrient in question also you label. So, you are having some ratio. Suppose I have a bound, I have iron 59 and mugenic acid or DMA, I label it with carbon 14, C14 DMA, and I know this is a complex is formed now. Okay. So, this complex will have a ratio of iron based on the energies. I showed you iron 59 will have a different energy. The slide, radioisotope slide, common isotope, this will have a different energy, carbon 14 will have a different energy. So, I can find out in my developed iron 59 C14 chelate complex, the ratio of the two energies is this. Okay. So, if you find in the plant, then you trace it in the plant. If it is going together, the ratio will remain unchanged. If it is traveling taken up by, but in the arm, in my leaf, if the ratio changes, that means now they are not traveling together. You have more signal of A energy and less signal of B energy, that means they are getting dissociated. Till they are together, the signal or the ratio of the signal will be same. If the signal is changing, that means they are dissociated, and this is how they found out that this chelate is not only taken up as a bound form complex, but not only uptake is there, translocation is there, but even the translocation to the grain is occurring as a complex. So, this experiment I value, and you guys, it is a very important thing. This is also, we say root exudation, I have already told you, this is sap flow rate. Now, we are slowly deviating from the nutrition to the source sink. So, if the carbon is traveling, at what rate the vascular sap is moving, you can find out using the tracer. So, I have a tracer, and if I measure, I cut that and I measure the at one time what is it, second time what is it, third time what is it. I can measure the rate at which the sap is flowing in the vascular system using label. And simple thing is, either I expose the plant with C14 carbon dioxide, and then after some time interval I cut the twig, measure the root exudation, and compare two plant species under a stressful condition, be it any abiotic stress, what is the flow rate of carbon in the vascular sap, at what rate it is travelling from the root to the shoot, you can find out. This is autoradiograph. It is a technique because any radioactivity will be emitting the radiation, and if I expose it to an X-ray film, this X-ray film will have, like we have any X-rays, when you stand in front of a for X-ray, there is a X-ray film that will be placed behind you, okay, and you will be exposed to the radiation, and based on the differential density of the tissues, you will be having a mark. So, here also based on the differential radioactivity that is there in the plant, it will be, if the radioactivity is more in an area, it will be emitting more radioactivity, and that particular region of the film X-ray film will be exposed more rather than the area where the less of the activity is there. So, here a comparison has been made between bean and sugar beet under NaCl stress, and using sodium 22 they found out that sugar beet not only takes up the plant, takes up the sodium, but also translocate to the shoot, to the leaves, whereas the bean it only takes up but does not translocate to the leaves. So, this distinction also you can make out, and sugar beet is tolerant rather than bean. If talk about different metabolism, how we can use radioactivity to study different physiological processes, how we can study the source sink relationships. The most common isotope that has been used to study source sink relationship is carbon 14. Okay. So, we can use this. How we can use it? This is one example where the positron emission tomography technique has been used. You can very clearly, suppose I have a I want to trace the movement of sucrose. So, I feed the plant with sucrose. How I feed? The leaf is there. So, one leaf, what I will do is I will scrub it, I will apply little Vaseline under the leaf, I will scrub it, gently add activity of that, it is little bit little damage you will be causing, that is why scrubbing has to be very less. You apply it in the evening, not in the morning, because otherwise the sample will, the activity will get dried on the leaf surface without getting absorbed. So, you put it in the evening, okay, scrub the surface, add the radioactivity on it, the sucrose or any element of your, any compound of your study, and then trace the activity within the plant, how that element, how the pesticide, whether the pesticide, suppose I want to trace whether pesticide is taken up by the plant or not. So, what I can do is I can get labeled pesticide, apply it foliarly on the leaf, and then trace it in the plant system, whether it is traveling or not. And this is these are non-destructive techniques. PET is a non-destructive technique, very useful for studying the source sink relationships. This is another study where they showed, it is not mine, this has come in Frontiers in Plant Sciences. So, sometimes this is basically for the perennials. In the perennials, you have a bigger root growth, bigger canopies. You find one side of the tree will be drying, whereas other side is flourishing. Morning, Dr. Jambulkar gave an example how a very clear observation, or how an observing mind found out that branch on the pear which was not affected, and then the vegetatively propagated. So, this is also, you find some branches will be drying, some will not be drying. So, it just depends. That is why the application of the fertilizer has to be uniform throughout. So, if I apply the fertilizer on one side, assuming or thinking or believing that this fertilizer will be traveling to the other side of the canopy also in the root zone, that is not the case. This clearly showed using radioisotopes that the nutrients that are supplied on one side of the canopy will be available, vascular bundle is there, but these nutrients are only getting transported from the left side, those applied to the left will be transported to the left channel, those applied to the right will be transported to the right channel, and will be feeding the branches that are there on the left or the right side depending upon the point of application. So, all these observations, it is not possible if you do not use a radio tracer. This is the lady who did this work is sitting amongst you. She is Shamima, okay, one of the participants in your this thing. This is the work she did for masters. How, in this case, how we found out that how the iron, which is less mobile, is getting retranslocated, and whether there is an involvement of nicotianamine, which is a chelate, how the availability of nicotianamine will be helping in retranslocation of iron from the senescizing leaf to the grain or the productive tissue. And she worked under different nitrogen and iron availability conditions. So, very clearly you can see that the retranslocation is higher under the deficient condition rather than the sufficient condition. If I have sufficient of it, I need not resort to retranslocation. Retranslocation, because for retranslocation I would be needing a chelate. So, why should I invest in a chelate when there is already lot of iron that is available for the plant? So, deficiency, our response to the deficient condition or marginal condition will be different from that of the sufficient conditions, and these strategies are effective under less available condition or the low availability conditions. So, very clearly you can see if iron is deficient, you have a higher translocation of iron from the older to the younger leaves rather than the iron plus condition even in the presence or without nitrogen. This is another experiment I would just like to share with you, and in this case using C14 sucrose, how we found out the contribution of the stem reserves. Normally in wheat now we are all looking for a parameter, we are looking for a thick peduncle. We are looking for a thick peduncle. If the peduncle is strong, not only it will be resistant to lodging, okay, but also likely it is going to hold reserves, carbon reserves. So, what is the contribution of these carbon reserves that are held in the internodes and nodes and internodes? We tried to find out. So, what we did is, we know in the case of wheat, flag leaf is the chief contributor of the carbohydrates to the grain. Okay. So, in this case we applied the potassium iodide 0.3% to induce senescence of the flag leaf. So, when the flag leaf senescence is getting induced, we measured the chlorophyll, we find that it is declining. So, senescence is getting induced gradually in the flag leaf, and in the internode part, you know wheat is a hollow stem. So, what you do is you have an injection, a syringe, with syringe you draw a 10 cc of air and add in its place, you have created 10 cc of volume in the internode. Now, you add same volume, replace it with the same volume of radioactive sucrose. So, we add C14 labeled sucrose in the internode, and you trace the radioactivity in the developing grain. You trace the radioactivity in the developing grain coinciding with the induction of senescence in the flag leaf. As the senescence is getting induced, it is up to you, you can draw samples at the end of the 5-day phase, or you can take a sample as 1 day, 2 day, 3 day, 4 day, 5 day, have proper number of replicates, and you can find out, and with induced senescence or at the end of the senescence phase of the flag leaf, what is the total contribution of the stem reserves, and how that differs under the abiotic stress conditions or between the two lines, whether it is a tolerant line or a susceptible line. So, all these things are possible if we use radioactivity. So, this is another, we know the Rubisco, PEP carboxylase, these are all carboxylating enzymes. We can use C14 carbon dioxide or C14 radio tracer for measuring the activity of RUBPcase or PEP carboxylase. This is one example I just share with you, PEP carboxylase. This is regarding the dark CO2 fixation. We know PEP carboxylase is there in the C4 plants. C3 plants, the chief carboxylating enzymes is Rubisco. But we also found out that nodules, root nodules, they also have the PEP carboxylase. PEP carboxylase is there in the root nodules. So, we did in vitro and in vivo assays to measure the PEP carboxylase activity in the root nodules. So, what we did in this case, and I always tell you the experiment part also, the method part, because this is where you need to think. Morning Dr. Jambulkar told about the applications of radiations. He only gave you an overview of the applications that he is using or he can think of, but sky is the limit for such applications, and it is up to you You should know what is your target, and probably then if you approach him, he will be able to design a device and experiment for you guys. So what we do here is you have the nodules along with the intact nodules along with some root portion in the chamber. You take it in a chamber and you release C14 carbon dioxide in the chamber. For certain, you incubate it, co-culture, co-incubate the C14 carbon dioxide with the nodules of different varieties, and later remove the excess of the C14 carbon dioxide by adding KOH. KOH will absorb the carbon dioxide, unused carbon dioxide in the chamber. Once that is done, open the chamber, remove the nodule, crush them, dry them, grind them, and measure the C14 counts using the liquid scintillation counter, and you will know how much of the C14 is getting fixed, and this fixation is essentially coming from the PEP carboxylase. This also I love to share, probably this will be the last 2 slides. Why I share this is that these are the applications of this paper came in Frontiers in Plant Science. I am a part of it, and I feel very proud to say that we helped these guys publish this paper in Frontiers in Plant Science, because they were struggling to find a proof for this. They had developed the transgenics having nematicidal properties. So nematodes resistance was there, but they were not able to prove that nematodes are getting deterred because of the gene or the gene product that they have induced in the plant. So we gave them the proof. What we did is we exposed these plants with radioactive carbon dioxide. You can see the radioactivity has got incorporated in the plants. You can see these 3 plants, the autoradiogram is there by the side. B is showing the autoradiogram. So radioactive carbon dioxide is now in the plant. Now these plants, when they were co-cultivated with the nematode, and later you removed the nematodes from that, isolate the nematode, you find the plant will not have any nematode associated with them because of the gene that you have put in. But what is deterring the nematode? You had to prove that these nematodes are going closer to the plant, they are trying to feed the plant, they are trying to feed onto the plant, but if they take the first bite, they find some product which is not good for them, which is not palatable, so they come back. So we had to prove, then we measured the carbon, which is carbon dioxide C14 label activity, which is associated with the nematodes. So we showed J2 is a stage. Don't ask me, I am not a nematologist, but J2 is a stage which reflects the association of the radioactivity with the nematodes. So with this proof, when we approached the journal, they finally accepted the paper, and the paper went into publication. So this is one I wanted to share, irrespective, because it is, as I told you, I could have on my own never thought of this is a tool, but I can think of these ideas, these designs, these methodologies if you bring to me different problems. I'll share one very good example. One IIT student, he came to us, he was doing PhD and also a scientist in ICR Ranchi Institute. He came to us and says, sir, my palmitic acid is there, it is getting degraded, I want to know what are the products where this palmitic acid is going. He wanted to find out the products where this palmitic acid is contributing. Okay. The carbon, hydrogen of this palmitic acid alone, where all it is going. So what we did is that he had a chain of reactions. He is treating the palmitic acid through a series of तू मोबाइल देख रही बेटी, या मुझे तो पकड़ा दे. कौन सा वाला? मैं बाद में दे दूँ, भेज दूँगा, ठीक है. वो फिर वो डिस्ट्रैक्शन होती है ना, और प्यारी बेटी है तो डिस्ट्रैक्शन नहीं करनी. Let me come back. So this is done. So, what I was telling you about the palmitic acid story. So, what we did is for them, we bought the labeled C14 labeled palmitic acid. We made him run 2 series of experiment, one with labeled palmitic acid which he passed on through his scheme of procedures and came out with a product, and the unlabeled palmitic acid passed through the same scheme of procedure, same scheme of method or treatments and came out with the final product. And we had now labeled products, unlabeled products. We ran a TLC, thin layer chromatography. Okay. Both these products were run together on a plate, thin layer chromatographic plate, and we total run of that volume, we cut it into different zones. Say suppose the total is say 5 inches, so we cut it into 1.5 inches grids, and we scrub the gel from that, silica from that, and analyze the radioactivity in those portions. We did not touch the unlabeled lane yet. We only isolated the gel from the labeled part. This will give us for which of the grids that are having radioactivity. If we are having less of the radioactivity in one grade, more in the second grade, less in the third grade, that means these 3 spots are the ones which are holding onto a product which is emanating from the C14 of the palmitic acid. So, from this we know that where the carbon 14 of our palmitic acid is getting associated. Then once you have this information and you have a lane where unlabeled product is run, you cannot run this on the you cannot run it on the HPLC radioactivity, you cannot run it on HPLC, but we can run the unlabeled products on HPLC. Now we know which are the areas where the product is located. So, you scrub those gels and isolate, re-extract it in say petroleum ether or any of these products, solvents, and run it on HPLC to know which is the GCMS, sorry, run it on GCMS and measure the product that is there. So, this is also strategy we thought about it because when he came to us, otherwise we do not need, we develop methods, we develop procedures if the guys need it. Okay. So, this is another one which we did it for the horticulturists, this is for the perennials. So, you know, pruning is a thing that is being done, this is a main method that is being done by everyone. In the horticultures, they go for pruning of the canopy. If they do not prune, now how much of pruning is effective? How much of pruning they should do? So, and how much, if they are pruning, how much of the leaves are sufficient to fill or get optimized sized fruits on the plant. So, we did an experiment where we labeled the leaves that are proximal to the fruit, distant to the fruit, and saw whether the distant fruits they are contributing as source to the developing fruit as sink. Okay. And if we are having 2 fruits, one we are having a distant single fruit or 2 fruits, whether the carbon, the source sink relationship that is getting altered or not. So, this paper also has seen the light of, it is in public domain. I think this is the last one probably I have. This is an experiment which I was telling you, this has gone in the chemosphere, the paper has come back for revision, and this is regarding Sneha told about air pollution. So, I just thought I will add one slide about air pollution because it is not the discipline, it is the technology that is important and we should know, and my idea was to give an insight into how we can use these radioisotopes for different methods. This is here you have the dust particles, you have the suspended particulate matter, and this suspended particulate matter may originate from the vehicular pollution or from the dust or the fields, okay, and this will be having the heavy metals we are inhaling, okay, these whether heavy metals are getting absorbed by the trees. We have avenue trees by the roadside, So, whether the avenue trees, they are acting as a sink for these heavy metals that are associated with the suspended particulate matter. So, this experiment which we did with the, you can see different size of the particles and different particle load, which is all labeled with zinc 65, because zinc 65 is a major heavy metal that is associated with the. So, we use one. There will be other heavy metals. Our idea was if I use lead radio tracer, I can find out what is happening to lead, whether lead is being taken up by the plants or not. So, experiment was done with the mulberry leaves, mulberry plants, not the leaves, full plant we had. So, this we did, and we could find that this the dust associated heavy metals are really taken up by the trees, leaves, foliage, and not only taken but also translocated and retained in the stems, the bark portion. Okay. So, this means that these avenue trees are acting as a sink for the heavy metals that are associated with the particulate matter. So, these are the conclusions. Radioisotopes can be used to assess fertilizer use efficiency or nutrient use efficiency. I told you that you guys should think of how we can improve the nutrient use efficiency under both optimum and deficient conditions. Understand the mechanisms and the ionic interactions, assessment of the sink demand and the source capacity. You cannot increase the source, but you can alter how under different biotic or abiotic stresses the source sink relationship is getting altered, and it will give you a useful clue of the efficiency of the varieties, why a variety is susceptible, why it is tolerant. Okay. Autoradiography can be exploited to get insight. Real time isotope imaging without impacting the, it is non-destructive. The PET I told you, this is an old technique, we should all exploit it. Thank you very much.
So now the talk is open for discussion. We will take just one question.
Sir.
Please.
First of all, thank you, sir. From the basics till the end, I believe we must have not heard such a nice, brief introduction of the concept of radioactivity, which was so nicely explained by you. Up till now, I have not heard such a good explanation of radioactivity. And the question is, how would you like to contrast this radio labels with the fluoro labels?
Fluoro labels are different techniques, radio labels are different techniques. See, you can label fluoro labels, radioactivity, any element, nutrient that you can label. Fluoro labels, Ranjit can throw more light on it, you can label probably the proteins and all with that. But
we are using to label
proteins, but you cannot label the fluoro label a nutrient. But radio label, you can get radio labeled nutrient, any element of the periodic table. What if I want a radio label of, I am interested to study antimony. Okay, antimony, what is the, whether it is taken up by the plant or not. One is to find out the total. If I am interested to find out the source, what I can go to bark, I can go to Brit, tell them that I want to, what they will do is they will knock down a neutron from that nucleus, make the nucleus unstable, and they will have a radioactive antimony, they will give it to me, and I can use it to find out the source from where the antimony is coming into the plant system. So, this is my understanding, and I think these are two different domains. Radioisotopes have very distinct advantage over fluorolabels. Fluorolabels are useful if you are interested to study the proteins, but radiolabels are important. Correct me if I am wrong, but radiolabels are very, very important if you are interested to study the nutrient dynamics.
So, are there any challenges or darker side of radioisotopes also when you are using in this plant study?
No challenge. What is the challenge? What kind of challenge you think? Fears or something? Huh? No fears, because the activity I told you, 1 in 1,000, 1 in million, 1 in billion. You can detect it, so there is no challenge. The person you use is sitting happily in front of you. Sumedha also used it. So we have Pandu also used it. Okay. So I am happy that 3 of the guys who were associated with me are there in your class, and if they are happy, I think it has no effect. And I have been using it for the last 26 years. Eh? No, I have been using it. You can see, this is what I tell you. It is the activity, we are working with very low activities. And I also, this is to induce confidence in you guys. Alpha can be stopped by a thin sheet of paper or gloves that you are wearing. Beta can be stopped by a plexiglass sheet because the range to which it can travel in air is very less. Gamma we should be worried, okay, but the activities that we are using are very low. And as a RSO, radiological safety officer, we do have monitoring devices. Okay, we have TLD, thermoluminescent dosimetry, that are probably many of the guys who are working with radioisotopes, they are given those monitors. They will be putting those badges, just like I have this badge. They will have that badge stuck in the pocket, one on the wrist, like a watch. When you are working with radioactivity, whatever radiation that is you are getting exposed at the chest level or at the wrist level, it will be getting monitored. And every 3 months these are monitored and you get a note, and the permissible limit is 100 millisieverts per 5 years or 20 millisieverts per year. And how much we are using? We are using only 0.002 millisieverts per year. Okay. We are 0.002 millisieverts as compared to 20 millisieverts, that is permissible as per the international regulation. So there is no problem with the use of radioisotopes or radiation. But one thing it advises, if advised, if the cooker is hot, okay, if you are cooking something, if the cooker is hot, you don't keep touching the cooker till you feel your hand gets burnt. Okay. So the 3 things that we follow in the use of radioisotopes is time, distance and shielding. Time, you should spend minimum time in front of the radioactivity. It is not that I need radioactivity, I keep it here, I keep moving in that. When I need to use that as a for inoculation or for a assay, I remove it from the fridge, take it out, thaw it, add it, put it back. Time, distance. If I can work like this, I don't have to bend myself, lean and see what is there in that. You can't see radioactivity. So the second thing I wanted to tell in the morning is, if you are getting a radiated product is not a source of radiation. This Dr. Jambulkar failed to mention it, but if you are radiating something, it will have some changes at the metabolic level, okay, but it will never ever become a source of radiation. So you should not worry about it. So these are tools which we need to exploit. Don't bend on it. And third is use proper shielding. If I am wearing this sweater, it will cut the radiation. Why I should worry? Okay. Thank you. Thank you.
So by seeing your face, I can easily make out that you thoroughly enjoyed this talk. And let me tell you, we are also the beneficiary of Sir expertise. So when we were working on Rubisco activities, so along with Sir, we developed first modified method for the estimation of Rubisco activity using radioisotope. And there is a difference between Sir and other scientists. If you ask me that I want to use your facility, I'll tell you, okay, please go and use. If you ask Dr. Bhupinder Singh sir that I want to use your facility, he will go along with you. He will tell you what are the basic principal methods, how to use this, and maybe he will try and he will show you by doing also. So this is the best part with sir. He is a very prolific orator. I think we all are party to this. And overall, he is a very good human being also. So with this brief note, now I request my coordinator to please felicitate sir with a memento.
Thank you very much.
So your talk was like a treat for all of us. Thank you. Thanks for coming and accepting our
Okay.
invitation.
Any questions anytime after this schedule also, you are most welcome. I only tried to give you an overview. Okay. I think today's forenoon was a treat for you, should be a treat for you guys, because you get exposed to an area which was not covered by others in the presentation. This training should be unique for you because of the coverage of the radioisotopes and radiations too. Thank you very much.
Thank you.